Hello and welcome back to proteins. How do they work and what can happen when they're made incorrectly? First off, what even is a protein? Well, proteins are long chains of amino acids that fold into a unique shape due to chemical forces between those amino acids. There are 20 different amino acids that can be formed into proteins, and there are thousands of ways these amino acids can be combined. For example, there are about 2.4 quintillion different combinations for a chain of 20 amino acids with no repetitions. Proteins aren't actually limited by a length of 20 amino acids, and they can repeat amino acids as many times as necessary, so in reality the number is much, much higher. The sequence of amino acids and consequently the shape is determined by genetic instructions. That unique shape of the protein is extremely important because it determines the unique function of the protein. Different shapes of proteins can do different things. Membrane transport proteins are embedded in the cell membrane and are used to transport substances into or out of the cell. Hemoglobin is a protein in red blood cells that transports oxygen around the body. Enzymes are proteins that are used to catalyze reaction in a cell. One such enzyme is protease. Protease is used to break down other proteins. All these proteins have unique shapes that determine their job. But what happens when the protein is, well, wrong? Sometimes, proteins can fold wrong where the instructions from your genes can be translated incorrectly. In these cases, the protein will not function as desired. In some cases, the protein can become actively harmful to the cell. We are going to focus on one such example, prions. Prions are distorted membrane proteins in brain cells, and they are protease resistant. Remember protease? This is where it comes in. Proteases break down ineffective proteins, among other things, but it can't break down these prions. So the cell is stuck with a protein that doesn't work and it can't get rid of it. Even worse, scientists hypothesize that the distorted protein can bind to the normal form of the protein and distort it as well. It's kind of like a zombie apocalypse. You got one zombie. If this zombie bites two people and every subsequent zombie also bites two people, you will very quickly end up with a noticeable zombie problem that's sharply turning towards apocalyptic. The big difference is that you can fight off the zombies attacking your house. You can't fight off prions. Your immune system can't recognize anything as wrong because the prions are something created by your cells, so even as your brain is shutting down because of the buildup, your immune system remains helpless to do anything about it. Now, why do we care? I mean, statistically speaking, prion diseases are 1 in a million. Well, for one thing, there's no cure, and for another, prion diseases have a 100% mortality rate. Being diagnosed with a prion disease is a death sentence. Aside from the human impact, prion diseases can be very dangerous in animal populations. Scrapie is a prion disease that is unique to sheep and goats, and bovine spongiform encephalopathy, or mad cow disease, originated in cattle and can infect people. There was an outbreak of this in Britain in the 70s and 80s, and aside from the human cost, farmers would lose their entire herd if even one cow exhibited symptoms. Chronic wasting disease infects deer, elk, and moose, and is in wild populations. CWD is difficult to regulate because of that. In addition, prions have been shown to bind to plants and have been shown to remain viable. This makes it even harder to manage the spread of CWD in the wild. Despite being incredibly rare, these diseases are extremely dangerous, largely because we don't understand them very well. If there were to be another outbreak like mad cow in England, the losses could be devastating.